Hello, class. Today I will be talking about the nucleus, the rough ER, smooth ER, cell membrane, centrosome, ribosomes, mitochondria, the vacuoles, and lysosomes. So basically, just the animal cell, to be honest. So first I want to start off with the nucleus. I'm gonna use red real quick. So here's the nucleus right here, and just want to talk about what it does and what it can do. Is a central organelle in a eukaryotic cell that contains the cell's genetic material, which is DNA, or the tiny dense center of an atom. Uh, it acts on the cell's control center, while the atomic nucleus is composed of protons and neutrons, and it is also held together by a strong nuclear force. Next thing I want to talk about is going to be the smooth ER and the rough ER. So, the rough ER, it is it's this pink thing right here. It looks very rigid. They both look rigid, but the rough ER just contains ribosomes on its surface and is for protein synthesis and modifications. But the smooth ER is it just lacks ribosomes, and it's for lipid synthesis, detoxification, and calcium storage. The functions of the rough ER are to synthesize and fold proteins. They perform quality control on proteins. They transform proteins to the Golgi apparatus and vessels, and it's involved in the information of lysosomes and the synthesis of phospholipids for cellular membranes. For the smooth ER, the functions are synthesizing lipids, phospholipids, and steroid hormones. It also metabolizes carbohydrates, detoxifies drugs and poisons, particularly for abundant in liver cells, and stores calcium ions. The next thing I want to talk about is, did I not mark the Golgi apparatus? Are we serious right now? Well, I'm gonna draw the Golgi apparatus. Just give me a minute, y'all. Gotta look up a picture so I can do this real quick. It's not gonna be in the right place, but it's alright. Golgi. We're just gonna put app because I don't got enough space. Okay, so the Golgi apparatus is a cell organelle that modifies, sorts, and packages proteins and lipids into membrane-bound vessels for transport to other locations in the cell or for secretion. There's a lot of functions, but I'm gonna just name off a couple. Protein and lipid processing, sorting and packaging, and synthesis. For protein and lipid processing, it receives proteins and lipids from the ER and further modifies them, such as through glycosylation. I forgot how to say that, but yeah. Sorting and packaging, it sorts the processed molecules and packages them into vessels for transports, and it takes them to their final destination, such as a lysosome, the plasma membrane, or for secretion from the cell. And for synthesis, in plant cells it's different, but it's the site for synthesis of complex polysaccharides needed for the cell wall, and it's also involved in lipid metabolism and the synthesis of molecules like glycolipids and glucose. Next thing I want to talk about is lysosomes. Right here. Lysosomes are a membrane organelle containing powerful digestive enzymes that break down waste, cellular debris, and foreign invaders. They literally, like I just said, break down excess or worn out cell parts and materials take up from the outside the cell. So yeah, it's basically just taking care of your digestive system on all costs. Next one would be the vacuole right here. It's a membrane bound as well. Stores water, nutrients, ions, and waste products in the cells. Yeah, that's it for that one. I didn't really have much on that one. And here goes the big one, mitochondria. And now we move to the plant cell. I was talking about the animal cell in the last part, but I think I had forgot to mention it previously. So, yeah. First I want to talk about the cell wall. It's just a provider, basically for the cell. Yeah. And then same thing with cytoplasm. It just provides structural support and maintains the shape that it's in, making the cells unable to move around and be messed up. Next thing I want to talk about is the mitochondria. I know I have Golgi apparatus at the top. Also, my fault. I meant to circle those, but I have Golgi apparatus right here, but I've already talked about it, so I'm going to go ahead and move on to the mitochondria. It's known as the cell's powerhouse. It's an organelle within eukaryotic cells, and it often converts food into energy, regulates cell death, manages other cell functions, and stores calcium. Along with the mitochondria, we have chloroplasts, which is found in plants and algae. They use sunlight, water, and carbon dioxide to make sugar, which actually is glucose, and oxygen. And the process of that is all called photosynthesis. Along with mitochondria, I just want to add to it, they're like tiny power plants in cells. They take in sugar and they break it down to make a thing we call ATP. So yeah. The next thing I want to talk about would be the ribosomes. They're technically just a protective barrier, and they separate the cell's interior from an external environment, and they control the passage of substances in and out of the cell. So, yep. Now I want to talk about peroxisomes. they break down fatty acids and the synthesis of phospholipids and plasmalogens. So yeah, they're this little yellow thing right here, and peroxisomes is right here, ribosomes is right here. I keep forgetting to circle them. I'm just talking right now, but yeah. And now we move on to the thing we like to call exocytosis. Exo means exit, and this is the diagram I have drawn up for the video. So first, from the nucleus, the DNA is copied to the mRNA. When it's leaving the nucleus, it goes to a ribosome, and the ribosome ends up going towards the rough ER while building proteins and traveling towards the Golgi apparatus. The Golgi apparatus sorts, processes, and packages proteins into a new vesicle, and so then the vesicle can carry the proteins inside the cell or towards the cell membrane. If they go out of the cell, they can start to fuse or could be released outside of the shell, and we like to call this process exocytosis.